In order to understand the T cell receptor and antigen recognition at the cellular level, I want to start with the B cell receptor and then move on to the T cell receptor. B cells recognize antigen because they have an antibody molecule anchored in the cell membrane, like you see here. Now, the job of this antigen receptor is to activate the B cell, trigger it to proliferate, and then differentiate to a plasma cell which will secrete the antibody, which will then bind the antigen and hopefully, by one of those effector mechanisms that I talked about, get rid of the antigen. Have it eaten by phagocytes, activate complement and get it degraded or something like that, or block it from binding to host cell structures. The problem is that the antibody bound on the B cell surface extends only a very short distance into the cytoplasm. There are only 3 amino acids on the cytoplasmic side of the antibody when it's anchored in the cell membrane, down here. Well, 3 amino acids in the cytoplasm is nowhere near enough to trigger any kind of signal transduction. So how are you going to get a biochemical signal to the inside of the B cell if the antibody only has 3 amino acids on the cytoplasmic side? It may bind antigen, which triggers a aggregation response to the antibodies, but it doesn't transduce any signal. So instead, the way it works is there are additional molecules associated with the antibody in a non-covalent fashion, which do extend way into the cytoplasm and are effective at transducing the signal. So it's called the signal transduction complex because it's a complex of multiple molecules. The antibody triggers that transduce the signal are called Ig alpha and Ig beta, and just because they're Greek letters doesn't mean they're part of antibody molecules. They're not. They extend well into the cytoplasm and have domains that activate protein phosphorylation or trigger tyrosine kinase activation and can trigger second messenger cascades. There are also additional molecules called CD19, CD20, and CD21, which also extend well into the cytoplasm and are also associated with the B cell's antigen receptor. So what happens is that when antibody binds to the B cell receptor, which is the antibody, it triggers them to agglutinate, or not agglutinate, but aggregate, which causes an aggregation of these associated molecules shown in the black. They then cross-activate each other, sending a second messenger cascade to the inside of the B cell and tell the B cell, hey, you just bound antigen, do something. Then it does something. What does it do? Well, it triggers the B cell to proliferate and differentiate to a plasma cell, which will then secrete the antibody. Now, on one B cell, there's not just one antigen receptor. Each B cell has about 100,000 of these receptor molecules on their surface per cell. But every antibody in those 100,000 receptor complexes has the exact same specificity for antigen. So one B cell recognizes only one antigen. It may have 100,000 receptors for that antigen, but it only recognizes that antigen. Now this brings up a problem, doesn't it? If each B cell can only recognize one antigen, one antigenic determinant actually, and you need to make over, maybe 100 billion B cells to recognize every possible antigen, because you have to make 100 billion antibodies, and each B cell can only make one of them. That would mean you'd have to have 100 billion B cells in your body. In your whole body, do you have that many B cells, do you think? What do you think? Yeah, you do. In fact, it's no problem. In fact, in every lymphoid tissue in your body, you've probably got 10 billion to 100 billion different B cells. Every lymph node has 100 billion B cells in it. There's plenty to go around. Every Peyer's patch in your gastrointestinal tract has over 100 billion B cells. It's no problem. There's plenty to go around. Every spleen in your body has 100 billion B cells in it, easily. How many spleens are there? 3, 4, whatever it is. There's 100 billion B cells in every one of them. There's enough completely to handle every possible antigen, no matter what route the antigen takes into your body. By the way, they don't let me teach anatomy. So that, now the CD molecules that identify B lymphocytes are CD19, 20, and 21. Fortunately, they go in numerical sequence, which makes them easy to remember. But they identify cells to a large degree as B cells, okay? So that's your B cell antigen receptor. Now T cells can also recognize antigen, but they got a completely different molecule called the T cell receptor. It has a lot of characteristics in common with antibody. It has a domain-like structure that look like antibody domains. 110 amino acids long, one interchain disulfide bond, forms little barrel-shaped structure. It has two chains that are linked together by disulfide bonds. We don't call them heavy and light like antibody because they're about the same size, so it wouldn't make sense. So instead they're called alpha and beta. T cell receptors also extend only a short distance into the cytoplasm, so they cannot transduce a signal by themselves. They have to have signal transduction molecules next to them. And for T cells, this molecule complex is called CD3. Now CD3 is a collective term for several different protein chains. As you can see, they're called gamma, delta, epsilon, zeta, and eta. And I don't show eta here. These are usually found as dimers, like the zeta chain dimer there. And they extend well into the cytoplasm, and they can transduce the signal for the T cell receptor. They just went down the Greek alphabet when they named these. So if the T cell receptor was alpha and beta, then they named the CD3 molecules gamma, delta, epsilon, zeta, and eta. The T cell receptor has variable domains, just like antibodies, and has a tremendous amount of variability. In fact, the number of possible T cell receptors ranges up to about 1 trillion possible receptor structures, more than you can possibly make in your entire lifetime. So there's a tremendous amount of variability in the T cell antigen receptors. Just like B cells, each T cell has 100,000 receptor complexes like this, but each receptor has the same specificity for antigen. So one T cell, one antigenic specificity. When the antigen binds to the T cell receptor, it causes a conformational change, which is sensed by the CD3 complex, which is next to it. Then it undergoes a conformational change and sends a signal on the inside of the T cell and says to the T cell, hey, you just bound the antigen, do something. And then it does something. And what it does in this case is it triggers the T cell to trigger some effector function, but there are many different T cell subsets, and they all do something different. So it depends on what kind of T cell it is, what that signal will actually trigger it to do. Now, there are some T cells that have different chains for their antigen receptor. 95% of T cells have an alpha chain and a beta chain, but it says up there also gamma plus delta. Now that's a different gamma and delta than the gamma and delta on the CD3 molecule. They got named approximately the same time, and that's where the reason for the overlap and confusion. But about 5% of your T cells have gamma and delta chains instead of alpha and beta chains. These are encoded for by different genes, and they do bind antigen. However, we understand these cells very poorly. We don't really know what they do. Gamma delta T cells are not found in lymphoid tissues. You don't find them in lymph nodes or spleens or Peyer's patches. You find them in submucosal regions, in the gastrointestinal tract and respiratory tract and skin. They have very limited diversity in their variable regions, probably directed against common pathogen structures of many pathogens that invade through mucosal sites. And we don't really know a heck of a lot about what they do. We shall never mention them again. Anything that anybody says about a T cell from this point on, for the rest of the course, they're talking about your standard, basic alpha beta receptor bearing T cell, right? Your standard issue T cell. with an alpha and a beta chain. And this shows, this is a nice picture actually, I like this one, of the T cell receptor complex, which shows the dimers of the CD3 subunits, epsilon delta or gamma epsilon or the zeta chain dimer shown in green, that actually triggers the signal transduction.